Bienvenidos a todos. Soy Giulio, soy ecologista de doenças italiano y desde 2012 trabajo en la Universidad de Stanford, donde estoy ejecutando un programa sobre dinámica y control de Schistosomiasis, que es ampliamente basado en la estructura de una salud, One Health. Infelizmente, eu realmente não falo português. Portanto, por uma questão de tempo, mudarei para o inglês, o meu inglês italiano. Peço desculpas por minha limitação. So, let me transition to English. And I want, first of all, to acknowledge the contribution of my large group of amazing collaborators who at different time and with different role contributed to formulate some of the concept ideas that I'm going to share with you today. There are too many to name them all, but I just want to briefly call out Dr. Sokol and Dr. Wood, with whom I have developed this entire program, Andy Chamberlain, our lab field manager who assisted us through all the activities. And because it is relevant for this context of the Brazilian meeting of Parasitology, I want also to remember my amazing Brazilian collaborator, Dr. Roseli Tuan, scientific researcher at the Secretariat of Health in São Paulo, Roberta Caldeira, research associate at Fiocruz in Belo Horizonte, Miguel Monteira, the National Institute for Space Research in São José dos Campos, and finally, also Thiago Pereira, research scientist at the Stem Cell Institute of Stanford University. With this audience, I do not have to remember that schistosomiasis is a debilitating parasitic disease of poverty endemic in 4 continents, in more than 70 countries in tropical and subtropical region, affecting more than 200 million people, the vast majority in sub-Saharan Africa, but as you can see here in the map, with the important pocket of transmissions of Schistosoma mansoni also in the northeast of Brazil. The important things for this meeting is that the parasites require 2 hosts to complete its life cycle, the definitive vertebrate host, potential people, and a limited set of freshwater snails, the intermediate host. So the parasite goes from people to the freshwater snails and back to people through these free-living water stages. With 2 free-living stages and an invertebrate host, all ectotherm organisms, no wonder that parasites' metabolism, its developmental biology, its mortality, the mobility, in general, the parasite performances, and ultimately the capacity to sustain transmission will all be affected by climate change and specifically by global warming. Okay, so I want to talk about that with reference to 4 specific points. The first one is how do we account for the direct effect of temperature on parasites and snail and what they mean for transmission. Then I want to talk how human response to climate change is going to affect the landscape in a way to potentially increase or decrease the risk of transmission of schistosomiasis. I also want to briefly discuss the problem of assessing transmission risk in the wider context of the ecological community in which the parasite is embedded. And last but not least, I would like to use the One Health framework to try to think about which type of solutions can we envision in order to have a better control of schistosomiasis. So let's start from the first point. So what you can see here is the thermal performance curve of swimming Circadia. Thermal performance curves has been derived to describe the metabolic, physiological, and demographic responses of both parasites and snails to temperature. This type of data allow to identify the thermal envelope, that is the range of temperature in that where snail and parasites are viable and when transmission can occur. And identified also, as you can see here, the critical thermal limits, that is the temperature that are either too cold, like here, or too hot to ultimately sustain transmission. This information can be used to map where transmission can occur, as I'm going to show you in the next slide. This graph here maps location where schistosomiasis is endemic according to their location-specific mean temperature, and specifically the mean temperature on the coldest quarter here on the horizontal axis and the mean temperature on the warmest quarter here on the vertical axis. On this graph here, we can overlap the experimentally observed critical thermal threshold for parasite intramolluscan development for Mansonae in orange and for hematobium in gray. We see that this critical thermal threshold delimit the space where schistosomiasis is endemic pretty well, especially for the upper critical thermal threshold here. Now, for each location, what we can do, as Dr. Stengsas has done in this seminal review paper published on Acta Tropical in 2019, is that we can basically project what's going to be the temperature in 2070. And when Stengsas did it, basically, the result is this huge shift to the right and up of all the locations that basically are going to experience warmer temperature both in the coldest period and in the warmest period. There are a couple of aspects here that are really interesting, and one is the potential expansion of schistosomiasis in area where it is not present, because all these locations will experience higher temperature, but also a potential range of contraction. There is all location whose temperature will be too hot for transmission. If we look at the projections in Stengert's paper, we basically can see that these are mostly the lowlands in the tropical belt, in the equatorial belt, where temperature is projected to become too high to support the intra-snail development of the parasites. And so we can see here it's going to be a range shift area where basically the risk is going to increase, or schistosomiasis is going to expand, at areas where the range is going to contract. Now, this is already a huge step forward, but increasing temperature is just one of climate change aspect, and how people, the economies, the countries, the society will respond to the challenges posed to climate change might also affect disease transmission, as I'm going to show you in these next slides. And although there is a lot to be said on this aspect, I want to focus on two specific points. That is what happened in terms of construction of dams and the crowding around dwindled waters, or the type of climate migration that will be induced, for instance, by drought. As far as the first point, there is no doubt that the expected agricultural expansion and intensification to feed a growing humanity, and also at the same time the need to fight climate change and increasing frequency and intensity of droughts in a warmer world, will require to basically develop more water management infrastructure. So more agriculture need water, and this water can be provided by dams. And schistosomiasis is a textbook type of example in which we know that basically the transmission risk is going to increase invariably following the constructions of dams. And there is a lot of literature that, can support basically this vision. And I want to show you with a specific example here. This is the Jama Dam in Senegal. The dam was built in 1986 at the mouth of the Senegal River, the border with Mauritania, to support agricultural expansion and prevent saltwater intrusion. It worked incredibly well, but unfortunately, the construction of the dam was immediately followed by what was probably one of the most spectacular outbreaks of schistosomiasis, as clearly documented by several peer-reviewed papers. Why that was the case? Well, the reason is connected to the massive landscape transformation that occurred following the dam, and that basically created suitable habitat for the snail that amplify the schistosoma parasite to thrive. As you can see here in this satellite imagery, 1985, 1 year before the dam completion, a pretty arid basically landscape. In 1999, 13 years after the dam completion, in which you can see areas that are much greener thanks to irrigation and to the permanent, basically a stable water. this is, that it's implying a cascading, basically, set of consequences that's been fully described in several papers of ours that we have cited here. There is something you can't see from the satellite imagery, which is this one, is the African river prawn, a native species of Western Africa, a voracious predator of the snails that amplify schistosomiasis. And it happened to be also a migratory species that require fresh water at the mouth of the Senegal River down here, but then is the species that swim upstream, potentially hundreds of kilometers for growing up and feeding, among the other things, also on the snails that amplify schistosomiasis. So when we built up the dam in 1986, we basically built up a big wall that prevented these upstream migrations, confined the prawn population downstream the dam, and extirpated completely upstream, thus removing a very important agent of top-down biological control of the snail population, as we described here, to creating basically, the perfect storm. Now, basically, we have rerun these analyses on several water basins where dams have been built, and we have shown that there is supportive evidence to basically believe that this happened over and over again through the continental Africa. And we have no reason, actually, to exclude that similar processes might be at play also here in Brazil. Although the climatic conditions, the lifestyles are completely different, most of the ingredients are actually here. Pockets of endemic schistosomiasis transmission, hundreds of dams already built through Brazil, hundreds, if not thousands of small artificial reservoirs that will be built in the next 2-3 decades to support agricultural expansion. A significant migration from northeast coastal zones where schisto has patches of endemicity to both the Amazon and the south of the country. Evidence from the literature, as reported here in this beautiful paper by Barbosa, of a connection between rice cultivation in northeast Brazil and increased risk of schistosomiasis. And last but not least, climate change that might make some of these habitats more suitable for the intermediate snail host. So the ingredients are there, and how they're going to interplay among each other is unclear. And in fact, I want to mention that this type of analysis face an additional level of complexity, specifically in Brazil. And this leads me to my third point, the wider ecological community where the parasite is embedded. And the reason is that, in fact, in Brazil, there are, there is 10 or more species of Bionfilaria, of which only 3 of them have been shown to be suitable hosts for human schistosomiasis, as reported here. How environmental drivers affect the outcome of the competitive interaction among these species is currently subject to investigation. I like to take the opportunity of talking to such a distinguished audience to mention that one of our side projects, in fact, with Dr. Tuan and Dr. Caldera, is to train machine learning algorithms capable to identify the genus and species of snails and parasites of medical importance by simply taking a picture, for instance, on a mobile phone or some more sophisticated camera in case that is available. We actually have a system that works surprisingly well in the lower basin of the Senegal River, where we only have to identify Bulinos and Bionfilaria pfeiferi species, but we want to expand these analyses to the Brazil landscape that, as I mentioned, is much more complicated. And if you are listening to my talk and you already have a set of digitized pictures of snails and parasites of medical importance, or you are willing to gather them, please contact us, because we would like to explore with you the possibility of expanding the collaboration and the training dataset. So this leads me to my latest point. It's about solution, and I want to mention that because when we are able to identify a problem, we believe that there might be also opportunities, at least, for solution. For instance, back in our field site in Senegal, by using a combination of lab experiment and field studies and modeling analysis, we show that the restoration of the native river prawn, for instance, through the construction of prawn ladders to bypass the dam, may contribute to reduce disease transmission. In addition, we also shown that the combination of medical treatment with prawn restoration is much more effective than just using medical treatment alone. It's something that should be considered as an opportunity of synergize and leveraging on the ecology of, the basically ecosystem, by restoring the native prawn. And in fact, there is more. Prawns are also delicious and nutritious food item that were part of the local diet in Senegal. We thus conducted a market analysis showing a great potential for developing prawn aquaculture in Senegal. Specifically, we identified that rice paddies are important transmission sites in the lower basin of the Senegal River, where we could actually develop integrated agriculture and prawn aquaculture. And through a modeling exercise, we show that prawn aquaculture could provide a source of protein for the local population, support small aquaculture business to fight poverty, while delivering an important public health service as described here, and in the case of prawns, restoring an important freshwater species. So what we call the 4 pillars of poverty alleviating, one health-based solutions. This led me to basically the end of my talk. I just want to mention that the One Health framework is extremely powerful, basically to identify the multiple pathway of impact of climate change on diseases with a complex life cycle. But I also want to encourage you to use the One Health framework to take one step forward and basically think how, by understanding the ecology of transmission and the potential role, for instance, of natural predators, we might be able to basically develop solutions that are able to target more than one sustainable development goals at a time, and so fight poverty, reduce anger, and improve health, and potentially restore an ecosystem integrity. And we have discussed this thoroughly in this paper that we just published a few months ago, and I suggest you to read. I want to conclude by basically thanking the agencies and the foundations that allowed us basically to do this work. And in case you are interested to know more, I want to suggest you this beautiful 10 minutes documentary that has been shot by the California Academy of Science, about our work in Senegal. And you can just Google protected by prawns and probably, find it easy. And it's a really nice descriptions of the work that we have done there in terms of biological control of schistosomiasis. And with that, I want to thank you so much for your attention. And I just can't wait to move in the question and answer session to discuss with you, the multiple challenges and the complexity of the problems that we're dealing with. Thank you very much.